Hello, this is Dr. Inarani Ray. I'm going to talk about horizontal gene transfer in bacteria today. Bacteria conveys their genetic material by two ways. One is vertical gene transfer and another is horizontal gene transfer. Vertical gene transfer means the transfer of genetic content through binary fission to the progeny. And horizontal transfer occurs through three processes: conjugation, transformation, and transduction. Conjugation is a direct transfer of genetic material between bacterial cells by direct cell-to-cell contact or by a bridge-like connection between two. In transformation, bacteria take up DNA from their environment and incorporate it into their genome. In transduction, movement of DNA occurs between bacteria, which is mediated by profiles. Today, we will discuss on the bacterial conjugation process. So, bacterial conjugation is a genetic recombination in which there is a transfer of DNA from a living donor bacterium to a recipient bacterium, often involves a 6-pilus. We can see the formation of 6-pilus here. The phenomenon of bacterial conjugation was discovered by J. Lederberg and E. Tatum in the year 1946. They took two mutant E. coli strain A, which could grow on a minimal medium only if the medium was supplemented with methionine and biotin, and strain B could grow on a medium only if it was supplemented with threonine, leucine, and thiamine. After mixing these two mutant strains, surprisingly, they got a new strain, which regained the ability to grow without added nutrients. From here, they concluded that probably a genetic transfer has taken place, genetic recombination has taken place, and that has been accomplished through bacterial conjugation. In another experiment, Bernard Davis constructed a U-tube, the base of which was separated by a filter. He kept the culture of each strain at each arm of the tube. Through the filter, media could pass, but bacterial cell could not. After mixing the culture media through pressure or suction, they did not get any new strain of E. coli. And from where he concluded that physical contact is a must, is necessary for conjugation and gene transfer through conjugation. Now we are going to talk about plasmid. Plasmid is extrachromosomal DNA. It's a circular DNA molecule that replicates independently of the bacterial chromosome. It can be transferred between individuals and among species. And it carries antibiotic-resistant genes. Precisely, what is F factor? It's a plasmid, of course, and A for fertility factor is a conjugative plasmid transferred from cell to cell by conjugation. A factor can act as an episome, that is, it can be inserted into the main bacterial chromosome or replicate independently as circular plasmid. The F plasmid is a low copy number plasmid, around 100 kilobase pair in length, and is present in 1 to 2 copies per cell. It replicates once per cell cycle and segregates to both daughter cells in cell division. Here we can see the bacteria where along with the main chromosome, it is having the plasmid, that is the F factor. So it is called the F plus bacteria. On the other hand, this bacterial cell, it doesn't have this F factor, so it is called the F minus bacteria. As the F factor is called the 6th factor, so this F plus bacteria can often regarded as a male, and F minus bacteria is often regarded as a female strain. The plasmid contains TRA and TRB loci, and TRA is responsible for pyelin and other regulatory genes. And these regulatory genes help in attachment of these two cells and for initiation of conjugation. Now, the conjugation process. This is a F plus cell. This is the plasmid. Now, it is conjugating with the F minus bacteria through a mating bridge. Now, the plasmid, it starts replication through rolling circle model, and the replicated part is being transferred through this mating bridge to this F minus cell. Now what happens? At the end, two F plus bacteria are formed. There are three types of conjugation: F plus and F minus conjugation, Hfr and F minus conjugation, and R plasmid conjugation. Now, F plus conjugation process. The F plus male has an F plus plasmid coding for a sex pilus and can serve as a genetic donor. The sex pilus adheres to an F minus female, that is a recipient bacteria. One strand of the F plus plasmid breaks as relaxase or relaxosome, create a nick in one of the strand. The sex pilus retracts and a breach is created, the mating breach is created between the two bacteria. One strand of the F plus plasmid enters the recipient bacterium. Both bacteria make a complementary strand of the F plus plasmid, and both are now called the F plus males, capable of producing a sex pilus. There was no transfer of donor chromosomal DNA. So this is how the F factor is going to the F minus cell to form two F plus cells. Now, Hfr conjugation. So what is Hfr? Hfr means high frequency recombination. High frequency recombination means it has the tendency to be transferred at a high frequency. Genetic recombination, in which fragments of chromosomal DNA from a male donor bacterium are transferred to a female recipient bacterium following insertion of an F plasmid into the nucleoid of the donor bacterium. It also involves a sex or conjugation pilus. Here we can see the F plus bacteria with F factor, and what happens, this F factor, this F plasmid, it combines with the main chromosome, and now Hfr is formed, and this Hfr is being transferred to the F minus bacteria. So an F plus plasmid insert into the donor bacterium's nucleoid to form an Hfr male. The sex pilus adheres to an F minus female, that is recipient. One donor DNA strand breaks in the middle of the inserted F plus plasmid. The sex pilus retracts and a breach forms between the two bacteria. One donor DNA strand begins to enter the recipient bacterium. The two cells now break apart easily, so the only a portion of the donor's DNA strand is usually transferred to the recipient bacterium. The donor bacterium makes a complementary copy of the remaining DNA strand and remains an Hfr male. The recipient bacterium makes a complementary strand of the transferred donor DNA. Now at the end, what happens? The donor DNA fragment undergoes genetic exchange with the recipient bacterium's DNA. Here we can see with the change in the color. Since there was transfer of some donor chromosomal DNA, but usually not a complete F plus plasmid, the recipient bacterium is still devoid of the F plasmid and it usually remains F minus. So, Hfr recombination, here the F plasmid is integrated with the main bacterial chromosome to form the Hfr cell. Now this Hfr is contacting with the F minus bacteria through mating breach, And what happens? A part of this Hfr, after replication, it is passing through the mating bridge to the F minus cell. And after interruption of the conjugation, what happens? A crossing over is taking place between the main chromosome, the main recipient chromosome, and the incoming chromosome part. Now we are coming to the resistant plasmid conjugation. Resistant plasmid conjugation is just like the F plus F minus conjugation, only the thing, instead of F plasmid, the plasmid which is coming is the R plasmid. R plasmid means resistant plasmid, that means it contains genes for resistance against antibiotic, resistant against heavy metals. So just like F plus F minus conjugation, it is occurring, and the R plasmid is coming to the recipient cell. So this is all about the conjugation. Next part will be coming soon. Till then, thank you.